Hello everyone, and welcome to our lecture, our final lecture of the module, which is going to be on the sample size. And when it comes to talking about the sample size, I really like this quote. It says that anybody can build a bridge that doesn't fall down, right? I can take my kids into the backyard and we can make a big pile of muck. It doesn't take any skill or ability that whatsoever to build a bridge. But it takes an engineer to build a bridge that just doesn't fall down. And the parallel here with the clinical trial, of course, is that we want to answer an important question. And we want to do so in a scientific and rigorous way. And the best way to do that, all else being equal, is to use a large study. But at the same time, we know that we're exposing patients to unknown risks. We are essentially asking them to give up their autonomy in treatment choices. And so while more patients is always better from a statistical point of view, from an inferential point of view, we also want to minimize the number of patients that we have coming into our trial. In other words, we want to build a great bridge, a study that answers the question, but we don't want to overdo it or make it unnecessarily large. We have another issue here, which often becomes the most important consideration, where the sample size just becomes some function of how much money you have at the start of the study. But ideally, we want to do these things in a more principled way. And that's what this talk is about. This talk is about how we set the sample size in a way that minimizes the number of patients we're going to use while maximizing our ability to answer the question at hand. And importantly, with respect to this module, it's a lecture about how you can talk to the statistician and the information they need from you in order to make this calculation. And so what usually happens is that people often come to the statistician quite late in the process, and they almost always start with the question of how big does my study need to be? And the reality is that how big the study needs to be, hopefully we've started to see this now over the course of the module, is a function of a lot of other different things that have to do with the study's design, and things that ultimately have to do with the research questions being asked. And so this is the reason why statisticians will always encourage investigators to get in touch early. Because if your assumptions and the study design and everything kind of that leads up to the sample size calculation, if there are mistakes there, then there's going to be a mistake in the sample size. And the sample size, it's important to say, is already a pretty fluid concept. It is an error-filled thing. We are making an educated guess about how many patients we need enrolled into the trial to give us a good answer. And hopefully that'll become clear here. So we really want to shore up everything about the design as early as possible, so we make sure that our educated guess is as educated as it can be. And to not take this seriously, is to not take the study seriously, to not take your questions seriously, to not take your patients seriously. So it's very important that you're able to talk about the stuff about your study design that's going to contribute to how you make the sample size estimate or calculation. So let's just keep up with a simple scenario that mirrors what we've been discussing over the course of the module. So we're going to have a 2-arm parallel trial, just so we can continue to discuss. And in this case, what we're going to have is a continuously measured primary outcome. And the principles involved in what we're talking about are pretty much the same when we're talking about continuous versus categorical outcomes. And I'll try to highlight that a little bit. And importantly, we're going to make things simple for calculations and say that these are equal sample sizes. So we've either gotten lucky or we've used some kind of a restricted randomization. So we have equal sample sizes in both arms. And that we know that maximizes our power and efficiency. And we're asking this question. The question we're always asking is treatment B superior to treatment A. And we want to know, to run this trial, how many patients do I need to enroll to make me feel more confident that we're going to get a good answer to our important question. And essentially what the statistician will need from you is 4 different things. All right, the first thing we need is the distribution of the outcome under the control condition. And what I mean there is, let's say we enrolled a bunch of patients and we didn't do anything to them. They're just patients that have come into the clinic, and the outcome is blood pressure, for example. What we need to know is what is the distribution of blood pressure in that group before we do anything, right? And of course, eventually what we're going to be doing is we're going to test the hypothesis that two different groups share that distribution, right? And if we see deviations, if we see one group, the distribution is different from that, then obviously we're going to conclude probably that had something to do with the effect of the treatment in the trial. So we need this kind of understanding of what would happen to our group of patients going forward into the future if we didn't do anything to them. And often for something that's continuously measured, the best we can do in terms of describing its distribution is to say what its mean and variance are. All right, so if we had blood pressure was going to be our primary outcome, then I want to know what the mean and variance of the blood pressure is in the clinic among the patients that you're going to be seeing and enrolling into the trial. All right, the next thing we need is we need to know what effect size you want to power the study for. And the argument we're going to make here is we want the smallest effect size of interest. And there are different ways of thinking about this, and Stevenson calls it an effect you'd like to not miss. And if you think of it this way in the context of drug development or developing interventions, you might test a number of different interventions to try to solve a particular problem. If you get to the end of a clinical trial of one of those interventions and you fail to see a difference between the two groups, you fail to see a treatment effect of that particular intervention, then it will probably disappear, maybe forever. Nobody will ever pick up that intervention again because they'll see, oh, someone already tried this, it didn't work. But it could have been that you just had a false negative, a type 2 error, which we'll come back to. And so you might be very remiss to miss out on a treatment that might have actually worked. And so what you want to make sure you do is you think hard about the smallest type of effect size you'd really like to see if it's in fact there. And people talk about this as the smallest effect size of interest or a minimum clinically important difference. But really importantly, what we're not doing is we're not trying to think about the effect size we think will happen, or we're not trying to base our studies on effect sizes that we see published in the literature. You really have to think about this and use your expertise. We'll come back to this. We'll come back to all these points. So that's the first two things. And then we have the type 1 and type 2 error rates. In other words, our thinking about our false negatives and our false positives and how we control those error rates. So that's four things that we need to talk about to do these kinds of calculations. And so then the question becomes, well, how do I know, right? This is all stuff that you need to bring to the statistician. The statistician will work with you, but this is stuff that, this is not a problem for a statistician to solve. These are the inputs that the statistician needs to solve a different problem. So how do you know? The answer is you have your subject matter expertise and your experience into the literature. You're looking at published trials and protocols. You're learning about the outcome. If there's one thing that needs to come through in terms of design over the course of this module, is how important outcomes are and understanding them. And we deal with this where people will come and they'll say, this is my outcome, and okay, well, how do you expect it to be distributed? Well, I have no idea. And well, you've been seeing patients for however many years, you've been measuring that thing, surely you can get some kind of sense of what the outcome is. And so really, becoming an expert in the outcome is an important job for a clinical investigator and an investigator in the context of a clinical trial. I want to make a quick note here. I've got the bottom there, not pilot studies. And what do I mean? Pilot studies often get misused as some way of kind of presenting preliminary data about distributions and maybe even effect sizes of an intervention if you do a pilot of an intervention. Generally speaking, though, pilot studies are too small to provide us with accurate estimates of those things. In other words, if I do a pilot study and I see of an intervention and I see a certain effect size, I then go off and I plan a definitive trial based on that effect size. Well, then why don't I just accept that effect size at the start? I don't know if it's an underestimate. I don't know if it's an overestimate. But the idea of building a larger study, more definitive study, and starting from that point, it doesn't make sense. And so what pilot studies and feasibility studies are really for is just kind of giving a dry run for all of your processes you're going to use in the bigger trial to make sure things work, to make sure instruments work, to make sure processes work, paperwork works, everything else. It's testing all the parts of a trial before you get to the main event. Let's break this down a little bit. Just as a reminder, we talked about type 1 and type 2 error. And so if we're in a situation, and often it's not this cut and dry, right? So this is just a useful fiction. If we're thinking about the results we see down here and then making conclusions, then if we see a result that says no effect, and the truth is no effect of the drug, and then we're correct. If we see that there's a treatment difference between B and A, and that actually reflects the truth, which we can never know for sure, that's our power. And then we have two types of mistakes. One where we see that there's a difference between the treatments, but in reality, there's no difference, and that's a type 1 error. It's a false positive. And then we have the opposite. We have the situation where there really wasn't a difference between the two things, but we didn't happen to see it in our results. And so that's a false negative, a type 2 error. And then everybody has their little ways of trying to remember what these things are. This picture tends to do it for me, with type 1 error on the left, a type 2 error on the right. A type 1 error is always a false positive. Now, an important thing about type 1 and type 2 errors, we're trying to maintain control of these, but there's always a trade-off between them. All else being equal, if I've designed the study, I've got the outcomes, I've got the sample size, I've got my effective interest. So if I take those other things that we have to have, and I don't change any of them, then I can always trade off between false negatives and false positives. And how can we think about this? If you think about your fire alarm in your house, it has a sensitivity to it. And it could be that the fire alarm is so sensitive that it goes off all the time. It gives you lots of false positives. It's constantly chirping at you and driving you up the wall. And so then you turn the sensitivity down and you make it harder for it to detect a fire or to give off the signal. And so the false positives go down, but now you're slightly increasing the risk of a false negative, the chances of actually missing, right? So there's always this trade-off. And generally, we've come to accept that there are kind of standard levels of these types of things. And certainly if you're doing a regulated trial where pretty much everybody would always expect the type 1 error rate, the false positive error rate, to be held at 5%. Right, so that's standard. Now, you could make a reasoned argument why it shouldn't be. But generally speaking, you're going to settle on this. We're going to control the type 1 error rate at 5%. And so then that leaves the power, or the type 2 error rate, and the power is 1 minus that. For the type 2 error rate, people generally use 0.2 or 0.1, meaning a power of 0.8 or a power of 0.9. And again, there's more leeway here for you to make decisions about what power you want. And generally speaking, I'd say that powering studies to 80%, where you'd have a 0.2 false negative error rate, is too low. But what winds up happening is that we hit constraints in budget and recruitment, and so this is where things tend to take a hit. Right? Sometimes you get stuck with the sample you've got, and that gets reflected in a lower and lower power. So what we want to do now is we want to put all of this together, and we want to break down this kind of picture of all the different parts here. And so what do we have? So the first thing we want to do, we talked about this in our earlier lecture on statistical inference when we talked about Name and Pearson. We want to just focus on this curve, this distribution. And so what is it? Remember, this is our sampling distribution under the null hypothesis of no effect. So X here is the difference in means. All right, so this isn't mean 1 and mean 2. That's not what this is. This is a distribution of a difference in means. I've subtracted the mean blood pressure in group B from group A, and I've gotten a number. And under the null distribution, our effect size would be 0. There's no difference between the two groups. All right, so that's number 1. And then we talk about this idea of a sampling distribution over and over again, which again is just a fiction, a useful fiction. It says that if I were to repeat the study an infinite number of times under the exact same conditions, which of course is impossible to do, it's a mental experiment. If I were to do this, and I met these certain assumptions, which is usually that the data are independent from one another, that they're identically distributed, the observations all come from the same distribution, then I would see this probability distribution of values. Right? So then the peak of this curve and the bulk of the space underneath the curve all hovers around 0. So every time I did one of these hypothetical repeat experiments, most of the time I get a value that's close to 0. But sometimes I get a value, that's more extreme, just because of sampling, right? Just because of random sampling error, right? That is our sampling distribution under the null hypothesis. And that's going to be determined by two things, which is the width of this distribution, the standard deviation of that distribution. We call it a standard error. A standard error is just a standard deviation. It's a standard deviation of a sampling distribution. Why they give it a new name, I have no idea. But that's all a standard error is. And to get the standard error of our sampling distribution, it's some function of the variability of the outcome itself. So here, it's a sampling distribution of the difference in means of blood pressure, but the standard deviation of that distribution will be a function of the standard deviation of blood pressure itself, and then divided by usually the square root of n. All right, so what happens in terms of if I start to modify these things? If I increase the sample size, n, then this thing goes down. And so my standard error here, which is the standard deviation of our sampling distribution, becomes more narrow, right? This shrinks in, so it's not so wide. If I take an outcome and I use a lower variability outcome, then same thing. If this comes down, the standard deviation of the outcome comes down, then the whole thing comes down, the standard error comes down. And again, this curve narrows, right? So we have less variability in our sampling distribution. So that's step one. And then we set some critical value, which is this line, where we have our type 1 error rate. If we're using a two-sided test, so let's say we have a type 1 error rate of 5%, we're going to use a two-sided test, which means we're going to split that 5% into the two tails, because we don't have necessarily a reason to think that the new treatment will be lower or higher than the existing treatment, and we're hoping it's better, whatever that is, but we don't know up front. So we're going to split that 5% between the two tails of this sampling distribution. So then this bit here would be 2.5%. So we set this line, and we call it our critical value on the x-axis, where it separates 2.5% of that sampling distribution from the other 97.5. And that's where, if we saw a value higher on that than that critical value, that's where we would declare we would reject this null hypothesis. If we saw a value greater than that critical value, that's where we say, you know what, that's an unusual finding given the null hypothesis, the null model, and so we're going to reject the null model. Right? And so that is the starting point of how we do this. Now, what is this other distribution? The other distribution is what we're going to call our alternative hypothesis. It is centered at our smallest effect size of interest, or the minimally clinically important difference. This is our proposed effect size that we want to make sure we absolutely capture. And so this distribution is exactly the same as this one. It's just shifted over so it's centered on the effect size here on the x-axis, whatever that number would be. And importantly, I'll have set the power in advance, right? So the power here in this particular example might be 80%, which means that 20% here of this distribution falls to the left of that critical value. So if this was the truth, this would be the sampling distribution under this hypothesis, and 20% of the time we'd get a value of x that was to the left of this line, even if this was true. But if I get a value to the left of this line over here, I'm going to not reject the null, which is essentially the same as rejecting this hypothesis. So if the truth was this, and this was our sampling distribution, and I found a value here, then I'm going to reject this one. Right? And so you can actually kind of see here, if this is 20%, it actually looks like there's a lot of options there where you might have a true value that's this, and it's, wouldn't be extremely rare to find that just based on the sampling distribution, but you'd still reject it. And so that's why you have to be really careful about making sure that when you set this effect size that you're actually interested in, that you really are trying to get it as low as all of this stuff, for the most part, is set in stone, right? This difference in means, your effect size, you're going to set that based on some kind of rationale. Your type 1 error, alpha, 5%, that's usually not going to change. You're going to set your power, or your alternative, your type 2 error rate, in advance. So this line here, the fact that this overlap of this curve is 20% of it, isn't going to change. So really the only thing you can change for the most part is the sample size. And so you can work with the interactive app And you can see that as you increase the sample size, then these things narrow, the distributions of these things narrow. And what that happens is you're still going to have 20% of the distribution falling onto the left side of the curve if you've got a type 2 error rate of 20%. And so when this narrows, it pulls this distribution over this way, which means you have a smaller difference between the two hypotheses. You can detect a smaller effect size at the same level of power in type 1 error. And so that's why increasing the sample size is so important, right? The greater our sample size, the smaller effects we can determine, and vice versa. All right, so that's how all of this kind of fits together. And so go have a play with the interactive, and then make sure you get on and get any questions. This stuff can be a little bit confusing the first few times you hear it, but the important part is to go back to that list of things, the things you need to work with the statistician who can sit down with you with a graph like this and talk through it some more. And those things are this distribution of the outcome, which is going to give us the standard deviation here. If I understand the distribution of the outcome, then I need the standard deviation from that. You're going to get the minimally important effect size, or the smallest effect size of interest, and that's going to play a feature here. And then the type 1 and the type 2 error rates. And so if you can think through all those things, if this is a bit much, which it can be, you're fine. Just if you have that other information, then you go and talk to a statistician. Now, from a practical standpoint, you have to think about increasing power and think about things we can do to improve. And so, the easy answer is always to recruit more patients. And this is overwhelmingly where trials fail. Trials fail because they don't get enough patients. And we get to the end of trials and we get answers with very wide confidence intervals, very inconclusive answers because we didn't recruit enough patients. So at the feasibility stage, at the pilot stage, one of the most important things you can do, and if you're trying to get money to do a trial, is to demonstrate your ability to recruit. You have to be able to show. And there is this kind of pressure for people to do studies and run trials, and you get trials where people know that they're not going to be able to get enough patients, but they run the trial anyway. And it's a bad idea. And people should stop doing that. And I understand that there are pressures and concerns, but this issue is a really important one. So before you do a trial, if you're taking your patients seriously, if you're taking the questions seriously, you want to be really certain that you can get enough patients. And a lot of times what that means is that you need to find collaborators. And so this is why, the big trials, are always multi-center. And, because you might have, 50 clinicians all recruiting 4 or 5 patients over a period of time. And so if you're taking your study seriously, if you know that you can't get enough patients on your own, then that's when it's time to find collaborators and to do a multi-center study so that you can get your N up. We talked about covariate adjustment. And so again, if you do your homework early and you find that there are strong prognostic factors predicting your outcome, then if you can measure those things, without placing too much burden on the patients, then you should, because we've seen how when we adjust for covariate information, it explains away some of the variability in the outcome. It leaves less variability to be explained by the treatment. It reduces the noise, essentially, making it easier to detect your signal. So covariate adjustment is a very good idea. We also saw early on not to categorize outcomes if you don't need to. So doing a study of continuously measured blood pressure, where I'm going to look at the average of treatment effect as a function of the mean difference in the two groups, you will get much more information and require many fewer patients than if you do a study where you're trying to look at a difference in the prevalence of hypertension. So don't unnecessarily categorize continuous things. Doing so is absolutely a waste of patient information. And finally, it also points to the fact that we need to retain the sample that we get. And so often what happens is that you have no problem recruitment, but then we have a hard time retaining patients in the sample. And so ultimately, the sample size drops and causes all kinds of problems with selection bias to boot. So it's really important to think up front as well about the things you're going to do to retain patients and the things you're going to do to help them avoid the reasons that they might want to leave a study. And again, this is one of those things where if you don't think about this up front, You invest all the time and effort into the trial, and then again you get to the end of it and you've got a result that's highly questionable because people haven't stayed in the study. Okay, so that is the lecture on setting the sample size. I've got the rubric up for the final individual project. There is a section there on the sample size, and I'll put up some links to where you can go to try to calculate some sample sizes. But for the purposes of the individual project, what you need to do, and which will be marked on, is providing the information that the statistician would need in order to make the sample size calculation. In other words, you won't be marked on the correctness of a sample size calculation. And so if your outcome, for example, is continuously measured and roughly kind of unimodal distributed, it looks like a normal curve, then you need to talk about the mean and standard deviation of that outcome. You need to talk about the effect size that you want to detect. You need to talk about the type 1 and type 2 error rate. If you have a categorical outcome and you're looking at a difference in the incidence or the percentage of patients in one group versus another, then you need to talk about the expected baseline risk of that outcome. So what percent of patients would normally experience that outcome if you didn't do anything? Say it's 8% of patients experience it. And then you need to say what percent of patients you would see in the active arm based on your smallest effect size of interest. So you would essentially come out, we normally would see 8% of patients have this. We're going to say the smallest effect size of interest is where we would see 12% in our active group. You come back to the statistician with those numbers and a type 1 and a type 2 error rate. But importantly, you need to justify those numbers. You need to justify why you're saying the distribution should look like this, and you need to justify why your effect size is the smallest effect size of interest. You have to convince other people up front that the decisions you're making are good and valid and justified. All right, as ever, if you have questions, get on the message board. I know it'll be a busy week with people wrapping up their projects. We will be on the message boards, late at night after the kids are in bed, checking in. Don't worry about emailing on the weekends until you get your projects done. Just feel free to get in touch, email, message boards, whatever you need. And thank you very much.